Welcome to the 2021 Virtual Vanderbilt Summer Science Academy. It's Friday, which means we're going to be talking about the MD-PhD path. And since we're so close to the end of the summer, we've been working through the steps of training. And right now, I am thrilled that we have two faculty members who are here at Vanderbilt who are going to tell you about their journeys today. So we're going to start today with Dr. Patrick Hu, who is going to tell us about his personal story and motivations for his path, and then a professional story along the way. So thank you so much for being here. We're thrilled to have you, and I look forward to your talk.
Thank you, Beth. Thank you very much for the opportunity to speak. I want to welcome all of you. I hope the summer has been, if you are here in person, I hope the summer has been rewarding for you. Let me first give you some context. So I was born 56 years ago in the Detroit suburbs. My parents were born in China, moved to Taiwan right before the Communist Revolution, and then came over to the States in the early 60s. When I was 8, we moved to Louisiana. In college, I had two research experiences. This is probably not what you're expecting to hear. Neither one of these research experiences was actually that fulfilling to me. And yet I am where I am. So the first experience was basically a summer research experience at the company where my dad worked. My parents being Chinese immigrants, not to culturally stereotype, but they were fairly frugal. And so since there was an opportunity for me to work in a lab while living at home, there was never an option to potentially stay where I was going to college to work in a lab that might be better suited to my interests. I'm very grateful for everything my parents did, but I would say their advice probably was not the best advice for me to be heeding. So I spent a summer doing research at a petrochemical company. It was not so fulfilling for me. And then I had one other research experience I decided to give another shot. So during the school year, I decided to join a medical school lab and work for the semester. This is a Google Maps showing you where my dorm was in college and where the lab was. And this is how long it would take to commute by bus. And so that's if the bus was there right when I got to the bus stop, right? Vice versa. Okay, so in lieu of taking advantage of many opportunities that would have been right here within walking distance, I decided to do this partly because the project was of interest to me at the time. There was an investigator at McLean Hospital who was interested in a family of proteins that he thought was involved in long-term memory consolidation. It turned out not to be. I didn't know any better. But this commute was actually a problem because it meant that I was able to spend less time in the lab than I would have liked, and I was wasting a lot of time on the commutes. And time is precious in college, as all of you know. So, the take-home messages from this early portion of my career is, number one, find good mentors. Okay, I'm sure people have been telling you that over and over. It is true. And mentors don't just have to be your research supervisors. They don't have to be the principal investigator. Obviously, that person, would be an important and could be an important and high quality mentor for you. But professors who have taught you in a class can mentor you. I would say more importantly, near peers and peers can really function as effective mentors. And so near peers would be students or recent college graduates who are slightly ahead of you in their careers. A great source of near peer mentorship is postdocs or graduate students who are working in the same lab you are. The vast majority of them are more than willing to talk to you about their pathways, their decision-making process, how they might have done things differently. So take full advantage of that. And finally, your peers. So people who are your contemporaries in school who are interested in doing the same sorts of things you are. Talk to them about how they're navigating these issues, and others can also be beneficial. When you're picking a lab as a college student, I recommend that you focus on the quality of mentorship. I think that's much more important than the topic of investigation. You at this stage basically just want to learn how to do good experiments. And what you end up investigating as an independent PI is going to depend much more on what you study as a postdoc, not what you study now. So basically, I would argue that you want to be in a lab where you have high quality mentorship, whether it be from the research supervisor or from a graduate student or postdoctoral fellow with whom you are paired. And take practical considerations such as commute time into account. So these were my two research experiences. There was another pretty formative experience for me, and that is that I did poorly in school the first semester of my junior year. And this probably was the first time in my life where I had encountered any academic difficulty. And so this was a bit of a shock to my system. So this has to happen to everyone at some point in their career. And so this was the first episode for me, not the last, but the first. And this caused me to do some soul searching and to really question my motives for wanting to pursue what I wanted to pursue. When I entered college, I was pretty certain I wanted to go to medical school. I was less certain about research. I told you about my two research experiences, but doing poorly in school my junior year really led me to think hard about whether this is what I wanted to do for my life. When I was growing up, I happened to be a reasonably competent pianist and performed in local and statewide competitions. Coincidence or not, in my junior year, I also started dating a concert pianist. And so I began to investigate the possibility of a career in music. What I managed to secure was a fellowship that underwrote a year of study at a conservatory in Germany. This is where that was. What was fantastic about this fellowship is that there were no degree requirements. And so I basically studied piano with a professor at a conservatory, practiced a lot, but also had the freedom to travel a ton in Europe. And that's what I did. And that was enormously beneficial because I had a lot of time to myself and a lot of opportunities to really think about how I wanted to move forward in life. And this remains probably one of the most valuable and formative years of my life. And so at this stage, keep in mind that you are not defined by your academic performance, whether they be so-called successes or failures. We'll talk a little bit more about successes and failures pretty soon. I would urge all of you to consider the option of taking 1 or 2 gap years after college if, number one, you're not certain about what you want to do at the next stage. That seems to be pretty obvious. Number two, if you feel like you need a more immersive research experience than you've been able to get in college before you decide to embark upon a PhD or an MD-PhD program, then this is something that you should consider. I put an asterisk there because I think training takes too long these days, and doing a couple of gap years when you don't really have to do it, it's just adding unnecessary time. Now, I think in certain circumstances, it can be very useful. So for example, if you're in a situation where you don't have very many opportunities where you are to get a solid research experience, then this is something that can strengthen your application for MD, PhD programs or for graduate school. But I think if you're in a place that has opportunities, and you're getting good mentorship and good advice. You don't have to do this to become a competitive applicant for MD PhD programs. And I see that next week people are going to be giving you tips, so I won't belabor that point. Nonetheless, it is important to create time to actually think deeply about why you are pursuing the career pathway that you're pursuing. And my bad semester junior year, you really created the opportunity for me to do that. So I spent the year in Europe, came back, and started in an MD only program at the beginning. I was a first year medical student. I told you my research experiences were not terribly rewarding, but during my year had thought hard about why I wanted to go to medical school, and was thinking that being a clinically oriented MD focused on patient care might not be as fulfilling to me as I would want my career to be. And so when I started during my first year of medical school, I actually joined a lab. And unlike the lab at the petrochemical company where my dad worked, this actually was an academic lab doing bacterial genetics, and was definitely much more up my alley. I enjoyed that experience very much, and based on that experience applied to and was admitted to the MD PhD program as a first year medical student. So joined that program, and then began my research years in this lab. There's a recurring theme here. There are bumps in the road. Okay, so in this first lab, so as I became more experienced with research, I became more and more disillusioned with the research that was ongoing in this lab. And so after some discussion with the other two students in the lab and with some faculty members, I decided to try to switch labs. And so I spent 2 years and change in this lab. I had no publications from that lab, and this would be considered a quote failure. The MD PhD program was extremely supportive of me, and they said, look around, we understand the situation, and we're okay if you switch labs, and if it takes you longer to finish your degree, then we will continue to support you financially, so don't worry about that. And so that was fantastic. And so I joined another lab, and even though I'd spent over 2 years in this first lab without generating any papers, I did gain expertise. And so what that permitted me to do was to hit the ground running when I joined this second lab. And in this second lab, so unlike the first lab where I was kind of on my own, in the second lab, I was paired with an experienced postdoc. And so this was really a key to my quote success in this lab. I ended up completing my PhD in that lab with 3 first author papers and 1 co-first author paper. And so that would be defined as success. Now remember, so I'm the same person here in lab 1 and lab 2. In lab 1, things didn't work out. In lab 2, things did work out. Okay, and so I think the take home here is that one shouldn't take too much credit for one's successes, and nor should one blame oneself too much for one's failures, because there are a lot of things beyond your control that will influence your trajectory and ultimate outcome in any given experience. And I will mention again, find good mentors. So I think one of the reasons I actually ended up in lab 1 was that I decided to join that lab in spite of being told by more than one person that perhaps I should reconsider. So not only do you need to find good mentors, but you also have to be open to being mentored. And I believe that one shortcoming that led to my stint in lab 1 was that I was not open enough to being mentored and to consider other possibilities. So when you get to the grad school stage, talk to a lot of people when you're deciding on what lab to work in. Talk to people who work in the labs that you're considering, because they can tell you exactly what it's like to work in that lab, right? So the PI is just going to give you a song and dance routine, right? Because they want you to come work for them, but you want to talk to the people who are actually in the trenches. And also in graduate school, I want to put in a plug for junior investigators. So in spite of the fact that lab number 2, where I was productive, was run by a senior investigator who was extremely productive, I had the luxury of working directly on a daily basis with a senior postdoc. And so if you go to a junior investigator's lab, that will be a similar situation where you will be working in a smaller lab with someone who's actually just completed a postdoc not too long ago. And so that person's going to have expertise at the bench and is more likely, in my opinion, to be able to provide you with outstanding mentorship by spending more one-on-one time with you. And the last thing to remember is, so if you join a lab and things don't work out, that's okay. It's okay to change labs. It worked out well for me. And it happens more than you think it happens. And I make it an effort to try to be transparent about this to people that I mentor so that they understand that, it doesn't define them. It's not necessarily an issue with them. This, a lot of times there just isn't a good match between the mentor and the students. And if there are obstacles that can't be overcome, then a change of venue is in order. I will just run through the rest of my biography relatively quickly because this is some years down the road. So in 6 or 7 years, if y'all are in MD PhD programs and applying to fellowships in internal medicine residency, I will be happy to talk your ear off about our physician scientist training program here, which is one of the best in the country. But in any case, after I got my degrees, PSTPs did not exist yet. So I started a residency in internal medicine, did 3 years of that, did a fellowship in adult oncology, and as part of that fellowship, did a postdoc. I'll talk a little bit about what we're doing now, but we do C. elegans genetics. And then I got my first job in 2005 as a faculty member at the University of Michigan. You can do the math here. I was 40 when I got my first job. That's something to think about if you decide to pursue the MD PhD physician scientist pathway. I think training is taking too long now, and PSTP directors at a national level are trying to come up with ways to shorten that training period. So it is an issue because the length of the training does contribute to attrition from the pathway, but nonetheless, it's something to think about. Just as a qualifier, I have no regrets about what I've done and would do the same thing again, but you just have to realize that it will take time. And then about 5 years ago, I moved to Vanderbilt. And this is what I spend my time doing now. I spend about 2/3 of my time running the lab, supervising people who are working in my lab, and writing a lot of grants. We study aging and stress resistance, primarily in worms. We would like to also move that into mammalian systems, and I'll talk about that in a little bit. I spend about 1/5 of my time doing clinical work. I have an outpatient oncology clinic at the VA, a half day a week, where I see general oncology patients. And I am an attending on the oncology consult service at the VA for about 6 to 8 weeks a year. And then I spend about 2 to 3 weeks a year on the inpatient oncology services at Vanderbilt. And I also spend about 15% of my time doing administrative work. So as I mentioned before, I'm director of the PSTP in the Department of Medicine here. And I also work very closely with Chris Williams, the director of the Medical Scientist Training Program here, the MSTP. I am a member of the admissions committee, and so have some experience in knowing what to look for applicants. This is what we work on. So we work on a conserved insulin-like signaling pathway. These are just the core members of the pathway. PI3 kinase happens to be one of the PI3 kinase AKT pathway happens to be one of the most commonly mutated pathways in cancer. And this was the reason that I wanted to continue to study this after my graduate school. So this is a magnified C. elegans. It's about 1 millimeter in length. This is in adults. It's extremely simple. And the reasons to study it are the adults have fewer than 1,000 cells. So you can get very high resolution on functional experiments regarding which cells are doing what. The generation time is very short, and each gravid animal can give rise to over 300 progeny. And so that's great if you want to do genetics, because you can generate a lot of animals in a short period of time. The genome is small and gene rich. So this was the first metazoan genome that was sequenced in its entirety. So it's about 30-fold smaller than the human genome, and yet the worm has about the same number of genes. And so there's a lot less intergenic stuff, right? Which means if you're doing screens and isolating mutants, you're more likely to find mutations that are within genes that affect the sequence of the gene product. And then for those who are interested in development, the developmental lineages are invariants. This led to a Nobel Prize for Sidney Brenner, John Sulston, and Bob Horvitz. And they're transparent, which means that the microscopy is relatively straightforward. And the development of GFP as a reporter was first done in C. elegans. So a lot of good reasons to study this. And so why did I get into this? I was interested in PI3 kinase as a graduate student, and this is what led to my pursuit of oncology as a subspecialty. While I was a resident, one night I was on night floats, and there were no patients here yet. So I went to the hospital library and pulled out the latest issue of Nature, and there's a paper from Gary Rufkin's lab describing the cloning of C. elegans PI3 kinase. And so this piqued my interest, did some more research into it, reached out to Gary, and actually secured a postdoctoral fellowship even before I applied for my clinical fellowship. Fortunately, I was able to end up in Boston for my fellowship training. And then as I was starting my fellowship, I saw this paper from Gary's lab describing the cloning of C. elegans P10. And so, as you may know, P10 is one of the most commonly mutated tumor suppressors in human cancers. And so the intention was to study this pathway in the worm, take advantage of what you can do in the worm, learn new things about the pathway, and then move it into mammalian systems. Now, this has been a somewhat wayward path, and it has taken longer to traverse than I might have expected. But nonetheless, I'll just give you a quick example of one of the stories that have come out of my lab. And so we basically do genetic screens to identify new components of the pathway. In the interest of time, I will just move directly to this, and which is that in mutants that have reduced insulin receptor signaling, the animals will arrest as dowers. And the important thing for a geneticist is that these are easy to distinguish from these, right? Just in a light microscope. And so we've done a lot of screens just looking for animals that arrest as dowers when they're not supposed to, or animals that don't arrest as dowers when they are supposed to. And so from one of our screens that we've done, these are the genes which were mutated in the various strains that came out of the screen. The most recent mutant we've been working on is TRAP1 in the worm, which is conserved in humans. The human ortholog is called SSR1. It's involved in ER translocation. And so our work basically suggested that this molecule was affected insulin signaling because it's required for the biogenesis of insulin-like peptides. And so we have been able to move that into mice. And so we made global knockout as well as beta cell specific knockout mice to see what the metabolic phenotype would be. This is just showing you this is a global heterozygous knockout, because the homozygous knockout is lethal. Just showing you that at 6 and 8 weeks after starting male mice on a high-fat diet, the heterozygous mutants have impaired glucose tolerance. So if you give them a glucose load, they don't dispose of it as well, probably because they're not making as much insulin as the wild type animals. To summarize, find good mentors. That is the #1 priority for you at this stage of the game and moving forward, because if you do that, then you will have done something that I didn't necessarily do to the extent that I should have, and that will help you avoid some of the bumps in the road that I encountered.
Thank you so much, Patrick. That's really wonderful. I have to admit, let me get this. Here's my little C. elegans. I did, this is a giant microbe C. elegans. My graduate work was in C. elegans, so
Wonderful,
I'm
wonderful.
especially excited about that. I think we have time for 1 or 2 questions if anybody has any. I might have one. I'll give it a second to see if anybody chimes in. So it sounds like you first were pre-med and then you considered becoming a pianist and then went back to pre-med. Can you talk a little bit about that transition back to pre-med from music?
Yes. Yeah, so, part of this was, I would say, this is probably my cultural background that led me to continue down the medical path, which ultimately was a good thing. So, I talked to my parents about wanting to potentially pursue music, and, I had a serious conversation with them. And at one point during that conversation, my dad said to me, you're not that good at piano. So Asian parents can be blunt, and so that's what I was told. And he was right, right? Because at the end of the day, I wasn't good enough to make it as a performer, which is what I would have wanted to do. So I went ahead and applied to medical school during my senior year of college, even though I was still having some doubts. And that was helpful because, I got in, and so I had a spot before I went to Germany. And so that was, and at the end of the day, I think the parental oversight in this case was beneficial. In most cases, being a parent now, in most cases, it's beneficial.
Awesome. We have a couple questions in the chat. How do you feel that your passion for medicine and science compares between now and when you went into the MD PhD? And I'm not sure, I'm wondering if they're referring to the length of training and if that wavers.
My passion for science is greater now because I know more. And because I know more, I know how much I don't know. I mean, that's a cliche. And the other thing is that it's just exciting. So the work that we do a lot of forward genetics. And when you do that, what you're doing is you're mutagenizing animals and then looking for a certain phenotype, and you don't know what mutations are causing the phenotype. And so it's this black box. And so the feeling of discovery when you figure out what the causal mutations are, Beth, maybe, yeah, so you're probably familiar with this. It's incredibly exciting. These are always opportunities to discover new things. And so that, I would say that excitement probably wasn't there when I was younger because I wasn't, I just didn't have the big picture and wasn't doing those sorts of experiments. So it has, my passion has grown and continues to.
Great question. Thank you. Great answer too. Another one, how did you decide on your clinical specialty? And it sounds like it might've been connected to the research you were interested in too,
yes,
is that right?
It was 100% connected to the research because I was just very interested in the fact that, a organism that is under homeostasis can all of a sudden develop a tumor mass. And so what is causing that, right? And so that was really the focus of lab number 2 that I worked in, where I eventually got my PhD. That lab was initially involved in cloning an oncogene called B, which turns out to be a mutated form of the epidermal growth factor receptor. And so that's how they got into PI3 kinase. And so the work on it was clear that PI3 kinase at the time, so, we couldn't sequence tumors yet. So we didn't know what the role of PI3 kinase in cancer was going to be, but it was clear that it was associated with viral oncoproteins, and it was activated when cells were treated with growth factors. So it was completely a scientific interest that drove that decision.
Awesome. Awesome. Well, thank you so much for sharing all of that. So insightful. And I just really appreciated that the revealing of successes and failures. I think that is just often, or it's not said enough. It's sometimes hidden behind stories. When you see the polished version of science, it might seem like there aren't failures behind it. So thank you so much for sharing.
Correct. Yeah, not at all. Okay.
Awesome.
Thanks. Good luck to you all.
Check out our website to find recordings of all of our videos. We'll get them posted about a week after each of the presentations. We can't wait to see you!